Before you can understand DNA origami, you must first understand nucleotide base pairing. DNA strands are composed of structures called nucleotides, which consist of a deoxyribose, a phosphate group, and one of the four nitrogenous bases. These nitrogenous bases are adenine, thymine, guanine, and cytosine, and they bind with each other using hydrogen bonds. In 1949, Erwin Chargaff, through a series of experiments, recognized that adenine and thymine were always about equal, and cytosine and guanine were also always about equal. This observation led to the conclusion that we now know as Chargaff's rule that adenine only binds with thymine and guanine only binds with cytosine. The concept at the core of DNA origami was hypothesized long before anybody was able to achieve it, most notably by Adrian Seaman in 1982. In many papers throughout the 1980s, he spoke about how the consistency of base pairing in DNA could be harnessed to rearrange the shape. The method of creating these shapes, however, was not actually achieved until 2006 by Paul Rothemund with his famous DNA origami shapes. These shapes were created with the purpose of testing to see if the DNA origami method actually worked. In the following images created by Rothemund, these were the first time DNA origami was successfully executed, and in that same paper, the term DNA origami was first coined.
So you've heard a bit about how base pairing works, but let's talk more about why this is extremely important and useful when it comes to this new technology. The complementarity of bases makes DNA a very stable structure. The pairings ensure that the helix has a uniform width throughout, and the hydrogen bonds between the bases hold the strands together, tightly enough that they stay intact, but can still be unzipped when needed. In addition, the strands naturally curl into a helix, with around 10.5 base pairs per turn. We can then take a single strand of DNA and control where it turns and folds by adding a pairing strand only in certain locations. This is, at its heart, the essence of DNA origami. We can take a single, really long strand of DNA, commonly called the scaffold, and decide where we need to fold it in order to form a specific shape. Then, based on the sequences present at these locations, we design shorter strands, or oligonucleotides known as staple strands, as they staple onto the scaffold, with sequences that can pair with the scaffold and cause helices to form. Now that we have folds, however, we also want to ensure some additional structural stability. Researchers have found many ways to do this, but the most common is known as a holiday junction. It helps hold adjacent sections of the scaffold together by adding a complementary strand that binds to the first strand for a portion and the second strand for the rest, forming crosslinks. These help lock the DNA in a certain conformation. Theoretically, this is a great procedure, but realistically, it can be difficult to obtain a single intact scaffold strand of the length needed to form more complex structures. An alternative that has been used is the creation of tiles, or small pieces of folded DNA that have sticky ends that allow them to self-assemble into larger pieces of folded DNA. This again capitalizes on how favorable and specific DNA binding is, as these properties allow the tiles to connect and stay together, and also give rise to their ability to self-assemble. The last thing I'd like to touch on are some of the details of the actual designing of these strands. Where do the scaffolds and staples come from? How do researchers know what sequences to use, or if their design will actually produce the desired result? And the answer, as many of you may have guessed, is through technology. As DNA origami has developed, so have many programs and applications that allow us to visualize the design process and access databases of known DNA sequences from a variety of organisms such as M13 bacteriophages that we can use as our scaffold. In this next section, we'll show you a bit about how one of these softwares, CAD Nano, actually works.
The designing of shapes and structures for DNA origami are usually done through a program such as CAD Nano. You can download the program and try it for yourself. Instructions for Mac and Windows are available at douglaslab.org/tutorials. Let's take a look. The CAD Nano interface has 3 panels. You can hide and show them through the dock. In the first panel, you can add a number of scaffold strands that will eventually be connected. In the second panel, you can extend and edit those scaffold strands. You can extend the size of your workspace in the second panel by going to the edge and clicking on the orange arrow that appears. Next, I'm going to extend the scaffold strands all the way by option clicking at the ends. The Edit tool allows you to make connections between the scaffold strands so that you can eventually achieve one single strand for your shape. I'm going to go through and crop out a roughly square workspace. Next, I switch to the Erase tool so I can get rid of all the extra little bits on the sides. If the workspace is too big to be seen all at once, use the Move tool to shift your view around. Now, if you open up the third panel, you'll see this nice square base for us to make our shape on. Today I will be making a simplified smiley face. It's kind of like pixel art. I will go through with the editing tool and crop out regions I don't want, then use the Erase tool to get rid of them so I can have the smiley face shape in the end. Once we have our shape, we need to make sure that all the scaffold blue lines are one continuous strand. This can be once again done with the editing tool, which can be used to connect the different strands. If you'd like to learn more about using this software, you can go visit CaDNAno themselves on YouTube and watch their tutorials. Once you have one long continuous scaffold strand, you can use the Auto Staple tool to generate staples. These staples are what will hold the DNA in shape. Now, all the highlighted staples you see are either longer or shorter than the recommended range. If they're too long, you can go through with the Break tool and crop them. Once that is done, you're ready to add DNA sequences to the scaffold itself. Select the Add Sequence tool and click on the blue box end. Select any of the sequence options and you should be able to see a list of DNA bases that pop up. If you see any question marks or blanks like I did, that means your scaffold is not one continuous strand. Let's go back and see where we might have missed something. Now that we've fixed the mistake, we're going to try to Add Sequence tool again. There, all good. Once you're happy with your shape and added all your sequences, you can export your file as an SVG and go brag to your friends about how you created your own DNA origami shape. There are also pre-made shapes by CAD Nano that you can download and open on your own computer to view. Open up the CAD Nano interface, select the Open File on the bottom right, and browse for the file in your computer. Many of the shapes provided are building blocks that can be used to create more complex DNA origami structures, including nanorobots that Ozzy will explain in our next section.
While still being relatively new, DNA origami has been used in a variety of methods for experiments, medicinal means, and many other ways. One of the more prominent ways that DNA origami has been used is in drug delivery. In drug delivery, drugs are encased by the DNA origami structures. Next, these structures are injected into the bloodstream so they are able to reach their target cell. After this occurs, the DNA structure can open up, allowing for the drug to be released into the cell. One advantage that has been found with using DNA origami as the carrier is that many of these structures are readily absorbed by many types of cells. This hints at the idea of using DNA origami to increase drug concentrations in cells that would be much more difficult with other methods. It has also been found that triangular-shaped DNA structures can specifically target cancer cells. While still very preliminary, this discovery has shown the great potential that DNA origami has. Another way that DNA origami is used is in nanorobot and fluorescence studies. During these nanorobot studies, DNA origami and other nano-engineered products were used to create nanorobots. These nanorobots were able to move and carry nanoparticles such as gold or silver through designated paths. Additionally, it can be used for fluorescence, where the DNA origami structures will be used to bring different sections of a type of protein, creating fluorescence that can be detected by researchers. As experiments and research around DNA origami continues, more ways of utilizing the technique will develop as well as current methods being improved upon. The future of DNA origami is currently unknown, but the potential that the technique has is amazing already.